All right, hello everybody. Today we're going to be talking about chromosomes. We'll talk about sex-linked chromosomes. We'll talk about linked genes that are linked on chromosomes. We'll talk about chromosome mapping, and then we'll take a look at some chromosomal mutations and something called nondisjunction, which leads to the unequal distribution of chromosomes in gametes. That didn't work the way I wanted. Make that go away. There we go. Okay, let's get started. All right, the first thing that I want to talk about, I'll move my picture out of the way here, is called gene linkage. Some genes are inherited together because they are found on the same chromosome. They are linked. Take a look at the picture. The blue and the red chromosomes, the blue chromosomes, they have the genes A, B, and C, capital A, B, and C, all on one chromosome. So if you inherit A, capital A, you also inherit capital B, you also inherit capital C, theoretically. However, notice what happens during metaphase 1. They cross over, and so notice that the capital C gets separated from the capital A and the B. And so now the capital C is on the other chromosome. So it says, notice, however, that A and C are more likely to be separated during crossing over because they are further apart. Genes that are separated onto different chromosomes are no longer linked and can be inherited separately. So we can actually use that information. The A and the B are more likely to be inherited together because they're closer together on the chromosome, and they're less likely to be that the place where crossing over occurs is random. It could happen anywhere on the chromosome. So when genes are really close together, the odds that crossing over is going to happen right in between the two, between the A and the B in this case, is less likely than it is to happen somewhere in between the A and the C. So we can tell how far apart A and B and C are from each other, and we can create something that's called a chromosome map, and we're going to learn more about this later. So it says we can use the percent of time that two traits are inherited together or separately to determine how far apart they are on the chromosome. So if we're always getting gene A and gene B to show up together in the offspring, then that's an indicator that they're probably really close together on the chromosome. However, if on occasion A and C are separated from each other, that's an indicator that maybe they're further apart on the chromosome map. And so we'll take a look at that as we go here. Okay, next, let's just do a quick talk about sex linkage. Before I get into the notes, I just want to go over this picture, and that's because the College Board used an FRQ question a few years ago using the ZWZZ sex determination system of chickens. And that's a little bit, you guys probably aren't familiar with it, so I just thought I'd mention it really quickly. Obviously, in humans, the females are homozygous when it comes to their sex chromosomes. So they're XX and the males are XY. The Y chromosome is a very, very small chromosome, and it carries very little information. It basically gives the information for creating male genitalia, but not much else. The X chromosome is full of super important information beyond sex determination. And so it's a really important chromosome to have. If somebody were to be born without an X chromosome, that is not a survivable condition. If somebody were to be born without a Y chromosome, that is a survivable condition. All right, so just take a look. The grasshoppers have a slightly different system. In that case, the females are XX and the males just have a single X chromosome. In the roosters, they have a completely different system than what we have. So the males are the homozygotes and they have two Z chromosomes, and then the females have a Z and a W chromosome. And then finally, the bumblebees in this case, the females are diploid and the males are haploid. Do you need to memorize this? By no means. I just want you to be aware that there are different ways to determine biological sex in different organisms. Okay, so here's the question. Why do males show sex-linked traits at a higher rate than females? Males are more likely to develop hemophilia. They're more likely to develop red-green color blindness and any number of different sex-linked traits. The question is, why? So first of all, if the trait is carried on the Y chromosome, well, they're the only ones that get it. So the females aren't going to get any mistakes or mutations that happen on a Y chromosome. That's just for the guys. I got a tickle. Give me a second. All right. And then the females, what turns out for the females, so let's say a female is born, these are her two X chromosomes, and one of her X chromosomes is broken. She has a backup copy, so if this one's not working, this one might be working, and her body can rely on this one. The guys, they only get one copy of that X chromosome, so if there's something broken on that X chromosome, they don't have a backup copy, they automatically show the symptoms of whatever that genetic trait is. It could be just red-green color blindness, or it could be something like hemophilia. All right, and then just as an FYI, be aware, I say that the females have this backup X copy, but here's a weird thing. Early in development, every X chromosome in a cell becomes an nope. I said that, I wrote that wrong. It says every X chromosome in a cell becomes inactive. There's only one that is not inactive. So here's my point, sorry. Some people are born with multiple X chromosomes, and that's a survivable condition because they all inactivate except for one. And so even in a female that is just XX, one of her chromosomes becomes inactive, and we call that a bar body. So just be aware of that. Only one X chromosome in females is actually active in the same way that only one X chromosome is active in a male. All right, and then I just wanted to show you quickly to remind you how to do the Punnett squares for sex-linked problems. They are unique in that you have to make sure you show the sex of each partner, and this could be the ZYZZ system for the chickens, so make sure that you're paying attention to that. So the male would have an X and a Y chromosome, and in this case, the Y chromosome does not carry the trait, so you don't put a letter by it, and then the females are XX. Let me know if you have any questions about solving these. All right, next we're going to talk, there's a reason there's a fruit fly on all these slides. Fruit flies continue to be a really common organism that we use when we're studying genetics. It's considered a model organism, just like the pea plants were considered model organisms. They're super easy to grow. They have super obvious traits like red eyes or funny wings, or their body colors can come in different colors. They have antennae that are different shapes, different numbers of legs, whatever. There's all these different mutations and things that we can observe. They have a super fast generation time, meaning they have babies really quickly and they have lots of babies. We can purposely mate them. We can't do that with humans. So they're an awesome model organism. So we'll talk quite a bit about fruit flies, and we'll do a lab that has to do with fruit flies. All right, here's a puzzle for you. Researchers, Thomas Hunt Morgan, famous name, bred red-eyed females with white-eyed males. So there it is in the diagram. So see the red-eyed males and the red-eyed females and the white-eyed males. Well, getting them all turned around. All right, so then what did they get for results? Well, it says all of the F1 generation had red eyes. Okay, that must mean that red is the dominant trait. Totally sensible. But now, when we take these F1 generation and we mate them, if it was simple dimple, we should get a 3-to-1 ratio, red to white, and it shouldn't matter the sex. However, that's not what they got. When they bred the F1, all of the females were red. There wasn't a 3-to-1 ratio in the females. They were 100% red. And then the males were 50-50 white. And so red and white. So the question was, what's going on here? It says the F2 had the expected 3-to-1 ratio, but 25% of the females should have been white and 25% of the males should have been white. So what actually happened? Well, based on the name of this little section, my guess is you can figure this out. Okay, so what's the conclusion? The eye color gene must be carried on the X chromosome. Let's spend some time looking at this diagram. I think this is a very complicated diagram, but it's super important. So first of all, we've got 2 females, we got 2 males. We're just showing you their generation. Sorry, just that we've got these were the results from the original cross. So let's go back and take a look. So here's a female, and she has 2 X chromosomes, just like human females. And on these X chromosomes, she was carrying the wild type dominant red eye trait. So she's homozygous dominant. The male, it turns out, he had, it wasn't carried on the it wasn't carried on the Y chromosome, and it was carried on his X chromosome, and he had the recessive white-eyed trait. So it says, look at the diagram, W plus means wild type, which is the more common and dominant red trait, and little w without a plus by it means white, and that's the recessive mutant gene. The bent Y chromosome does not have the color gene. So look at the diagram and see that little bend, no color gene on that one. In the F1 generation, everyone looked red because they had inherited mom's dominant W plus. So here, this W plus came from the mom and this W plus came from the mom. These are the girl babies. These are the boy babies. They got their Y chromosome from their dad. So they got their W pluses. Everybody got their W pluses from the mom. Where did the girls get this lowercase w, the mutant trait? They got that from the dad. Dads give the X chromosome to their daughters and they give the Y chromosome to their sons. All right, moving on. Importantly, the F1 generation females were heterozygous as expected, but the males were not. We would have expected the males to also be heterozygous, but they only get one copy of this eye color trait. The Y chromosome doesn't carry it. The fathers did not have a color gene to pass on to the sons. So here's the fathers. They pass the color gene on to their daughters, but they pass the Y chromosome onto their sons. That's what makes them sons. So the fathers of the F2 generation always passed red genes on to their daughters only. So here's the fathers, and they passed their red gene onto their daughters, but they passed the Y chromosome onto their sons. The sons received a red or a white chromosome from their moms. So let that sink in for a second. So the dads are always passing on, in this example, the dads are always passing on the dominant red gene to their daughters. So all of their daughters were going to look red no matter what because they inherited the red gene from their dad. But the dads couldn't pass on any color trait, eye color trait, to their sons. So the sons are going to get whatever the moms had. The moms were heterozygous. So the mom sometimes is going to pass on the red gene and sometimes they're going to pass on the white gene. All right, so that explains those results. Okay, let's just glance at a sex-linked pedigree right now. This is specifically on the X chromosome. So it says, what are the genotypes of the following individuals? And remember, because it's on the X chromosome, you have to show the X and the Y chromosomes. So previously on pedigrees, we've done like big N, little n, or big T, big T. You don't get to do that now. You have to say X and then capital N or lowercase n, X, capital N, lowercase n, if it's a female, or you say XY and you tell me what the different, what's on the X chromosome, and then it's not carried on the Y chromosome. So let's go ahead and take a look at 1 and 2. So that's the first couple. The black colored in circle. That means she has the recessive trait. So when we're going to label it little n in this case. So the female would be X little n, X little n. That's why she has the trait. The male, number 2, does not have the trait. So he must have an X big N. And then his second letter is a Y. And what do we put next to the Y? Nothing, because it ain't carried on the Y. You gotta get that down. Okay, so let's look at person number 4. Person number 4 is a female, so we know she's gonna be XX, so you have to write the 2 X's down. She's normal, she doesn't have this recessive trait, so we're gonna give her 1 big N, and then we don't know what her second one is, but we look back at her parents. Mom will always give an X little n to all of her kids. Mom will always give an X little n to all of her kids. So number 4's second letter must be X little n. Number 4 must have gotten her X big N from her dad. Now, just as an aside, notice number 1's sons. She has 2 sons. They both have this trait. That's because dad didn't pass this trait to them. Dad passed his Y chromosome that doesn't have this trait on it. It was mom who passed that trait on to her sons. So that's why they have this trait. Okay, let's take a look at number 5. Oh, I just did it. Number 5, so it's a male, and he got the trait from his mom, so he would be X little n Y. And then finally, number 10 is a female. So we know that she is XX. We know that she has a capital N because she is normal or doesn't have this particular trait. She could have gotten that from either parent. The question is, what is her second letter? We know from the brother that they do do have this trait, and we also know that she was heterozygous. So we know they have that trait in their DNA. It's possible that they're both heterozygotes. It's possible that he is homozygous dominant. We don't know. So that means we know she got one X big N, but she could have gotten her mom's little XN, or she could have gotten little XN, X little n, I said that backwards, or she could have gotten a dominant one from her dad. So we don't actually know. So you have to put a question mark. We don't actually know her second X, what the which she inherited for that second one. All right, moving on. Now we're going to talk, that's all been sex linkage that we've been talking about. Now we're going to talk about autosomal linkage. Remember that the autosomes are chromosomes that do not determine sex. So the reason that matters, it sounds so boring, but the reason it matters is that males and females both get 2 copies, at least in humans and in fruit flies. They get 2 copies of both. So in the sex link, the males were only getting 1 copy of it. The females were getting 2 copies. Okay, so it says study the diagram. Number 1, the female is heterozygous. In this example, this is just an example. They mated a male and a female. They mated some. In this example, the female is heterozygous for body color and wing shape. So notice that she has a red gene that has the wild type because they have the pluses. And so that's the gene for gray body, which is dominant, and the gene for normal shaped wings. They're nice long wings. She also has another chromosome. This is what makes her heterozygous. She has a chromosome that has the gene for black body and vestigial wings, which are short little wings. The male that we're test crossing her with, that we're mating her with, has only recessive traits. So he's got 2 chromosomes, one from his mom and one from his dad. And they both carry the recessive black and vestigial wing trait. Here's the example of vestigial wings. The only way you would ever see vestigial wings is if the fruit fly got 2 copies of it. And it's just these little stubs of wings, and they don't really work. They can't really fly. So it's a bad mutation. Okay, so let's keep watching. So it says the male is homozygous for both traits. When the female's chromosomes go through crossing over during meiosis 1, so here we go. We're looking at crossing over. Crossing over happened in meiosis 1. The traits are separated. B is now sometimes with VG plus, the wild type. So take a look. This red sister chromatid, normal. So it didn't change at all. It's still B plus and VG plus on that one sister chromatid. But notice the second red sister chromatid has a little pink tip on it now. And that's because they crossed over with the other one here. These 2 crossed over and they traded VG genes. They traded wing genes. That's what VG is for, vestigial wing. So they traded those genes. And so now we've got a B plus, we've got the wild type gray body with a vestigial wing, a mutated wing, and we've got a black body with the full wing. So that doesn't happen all the time. We've still got the normal chromatids also, but we've had some recombination of those genes. Okay, because the male's alleles are homozygous, because he's just BVG the whole way, they don't even show him crossing over, because if he did cross over, which he did, it would just be moving 1 VG to 1 VG, and you would see no effect of it. So the VG of the first chromosome probably crossed over sometimes to the VG of the second chromosome, but it doesn't matter because no matter what, he's always got black and vestigial wings. He's not a heterozygote, so it didn't matter. Okay, so what does that mean in the end? That means that all of his sperm will always carry the black and the vestigial wing genes. So we've only got 1 gamete for him here. The female, however, has 4 different gametes that she could have. She could have the chromatid that was normal, the red one that didn't cross over. She could have the pink one that didn't cross over. She could have the red one that did cross over, and she could have the pink one that did cross over. So any of those can be fertilized by his sperm, and we're going to get different offspring. So it says the parental type offspring, we get some that are gray and normal because those were the dominant traits that the mom donated. We got some, we should have an equal number basically of ones that are black and have the vestigial wings because mom was heterozygous and had that trait just like dad had. And then notice there are some recombinant offspring. 206 have a cross. They have a gray body with the little wings, and 185 have the black body with the full wings. Those are recombinant offspring because during crossing over, the chromosomes, the genes recombined. That's what recombinant means. All right, so it says, look at the impact of crossing over on the offspring. Without crossing over, probability would expect a 1-to-1 ratio of gray normal and black vestigial, nothing more. So remember that the folks who were first studying this, they couldn't see these chromosomes. They're just mating them and they're making predictions about what the babies should be like. And so they figure they've got a dominant female and a recessive male. They should have a heterozygous dominant female. They should have 50% that look like the female and 50% that look like the male. But they didn't actually get that. So I'll show you the exact numbers in just a second. They got 390. Oh, the exact numbers are here. There we go. So they got 391 that didn't fit either category. They weren't the black normal wing, and they weren't the gray normal wing, and they weren't the black vestigial wing. They were a combination of the two. And so we're learning here, this is where they first discovered autosomal linkage and crossing over. So it says, however, there are now a number of recombinant offspring that they had not predicted. So look at the impact of crossing over on the offspring. Could the gray vestigial and black normal offspring actually be an indicator that these genes were on different chromosomes? So we're thinking like these scientists. They can't see the chromosomes. I gave you pictures of chromosomes on this other page so you could see them and knew what was going on, but these guys didn't know what was going on. So the question is, could the gray vestigial and black normal offspring actually be an indicator that these genes are on different chromosomes? Well, no, that wouldn't work either. So if they were located on different chromosomes, we would expect a 1-to-1 to 1-to-1 ratio. So for every gray normal wings, we would have, so if we had 10 gray normal wings, we would expect 10 black vestigial wings. We'd expect 10 gray vestigial, and we would expect 10 black normal. What a mouthful. Do you need to write that down? No. However, and that's if they're on separate chromosomes. If they're linked on the same chromosome, then we should see a 1-to-1 ratio. We didn't see either of those. Thomas Hunt Morgan, when he was doing these, he's like, where did these numbers come from? What's happening? Well, you and I know where those numbers came from. It came from crossing over. He didn't know what crossing over was. So now he's going to figure out what crossing over is, and we're going to create a chromosome map based on how often we're seeing these recombinant offspring. Notice that the majority of the offspring were not recombinant. They were exactly what Thomas Hunt Morgan had predicted. There's just a few of these that have crossing over. This is not mutations, you guys. This is crossing over. That's not a mutation. All right, so it says the actual results don't reflect either of the predicted ratios. So what are we going to do with this? So it says we can calculate the frequency of crossing over by looking at the numbers of recombinant offspring out of the total number of offspring. So look down at the bottom of the diagram. You've got 391 recombinants over the total number to give you about a 17% crossing over. So here's the question. What would happen to the recombination frequency if the genes were located closer together on the chromosome? Remember that where crossing over occurs on a chromosome is random. So B and G, pretend they're this far apart. What if they were actually this far apart? What would happen to that percent at the bottom of our diagram here? It's now 17%. Would it be higher? Would it be 25%? Or would it be lower, like 10%? The recombination frequency would be less because genes that are closer together are separated by crossing over less often. Let me say that again. My little thing was in the way. The recombination frequency would be less because genes that are closer together are separated by crossing over less often. So now we're going to create something that's called a chromosome map. We can use the recombination frequencies to make a linkage map of the chromosome. Genes that are further apart will recombine or cross over more often than genes that are closer together. Map units on one of these maps, so they actually call map units, like that's like an inch or a centimeter. They're called map units, and it's the distance between genes on a linkage map, and one map unit is equal to a 1% chance of crossover and recombinant offspring. So if you remember from our example that we had, and I put it here again, we had 391 recombinants out of 2,300 babies total. So we said we had a 17% recombination rate. So now we say that those two genes, B and VG, are 17% apart. 17 map units apart on the chromosome. So now if we were to just do some more experiments, we would expect, now it's never going to be perfect ever, but we would expect, and there's some reasons to it, and we're not going to go into it right now, but we would expect that, for example, this, I think it's called cinnabar trait. I'm forgetting at the moment, but the CN trait, we'll just go with that. We would expect that if it's between the B and the VG, that there would be less recombinant frequency between B and CN, and there would be less between CN and VG than there is between B and VG, which are further apart. And so that's how they place the CN in the middle of the chromosome map between the black and the vestigial traits. All right, we'll practice that a little bit more. So the last thing here, it just says the color and wing genes separated due to crossing over approximately 17% of the time. So those genes are placed approximately 17 map units apart. All right, totally changing gears here. Now we're going to talk about something called nondisjunction. Nondisjunction is when we have an error during meiosis, and it can happen in either anaphase 1 or in anaphase 2. So take a look at the diagram. Always study the diagrams. So remember that in metaphase 1, they're supposed to be lined up with their homologous partners. And then in anaphase 1, they're supposed to be separated from their homologous partners. But notice in the diagram that there's two that stay stuck together and they go to one side. And so what ends up happening, if you go all the way down, if you follow those two all the way down, you end up with some gametes at the bottom. On the far left, the two on the far left have an extra chromosome, and the two on the right have a missing chromosome. So it screws up the total number of chromosomes in the sperm or the egg or the pollen. Look at the other diagram. So the diagram on the right, if it happens not during anaphase 1, but during anaphase 2. Anaphase 2 is when you have bivalent chromosomes and they're supposed to separate from their partners into monovalent chromosomes. But in this case, in anaphase 2, if they don't separate from their partners, and again, they go to one side, what ends up happening? Well, two of the gametes are normal because they came from this set here, but two of the gametes have the wrong number of chromosomes. So we've got one that's got an extra chromosome and one that's missing a chromosome. If any of these gametes that have the wrong number of chromosomes fertilizes an egg or whatever, however you want to say it, or a sperm fertilizes, whatever, they're going to end up, the embryo is going to end up, the zygote, better word, is going to end up with the wrong number of chromosomes. Okay, so these mutations must happen in a germ cell as opposed to a somatic cell. Somatic cells are the cells of my body. Germ cells are the cells that produce our egg or our sperm. So they're at the top of the meiosis diagram, right here at the top of the meiosis diagram. That's a germ cell. That would never be a liver cell. That would never be a brain cell. That's a cell that's found in the ovaries, in the testes, or in plant reproductive structures, for example. So germ cells are the cells, that's what these are called, germ cells, you should know that word. Germ cells give rise to the gametes. They're the ones that divide and form the gametes. Every cell in the baby would have the wrong number of chromosomes if non-disjunction occurs during from one of the germ cells. However, if non-disjunction happens in me, say this cheek cell right now as it's going through mitosis, say its bivalent chromosomes don't separate from each other, but they stay stuck together. If that doesn't go through apoptosis, if my other proof checkers, I can't think what they're called. Anyway, if it doesn't get slated for death and it continues to reproduce, it would only affect the cells right here on my face because I've already got 100 trillion cells that are doing their own thing. The issue is when this happens in a baby that is only one cell big, or before the baby's fertilized, but when it happens in a baby that's only one cell big, and then it copies to make all 100 trillion cells, that baby has the wrong number of chromosomes in every single one of its cells. All right, moving on. So what are the consequences of this? Well, the consequences are called trisomies and monosomies. So in a trisomy, that means that they have 3 of a chromosome where they're supposed to have pairs. Remember, for every chromosome, you're supposed to get 1 from mom, doesn't matter the organism, 1 from the female parent and 1 from the male parent. In a trisomy, they got 3 chromosomes. In a monosomy, they only got 1 of the chromosomes. And let's back up for a second just to make sure you understand why. If let's say these are sperm, just for argument's sake, let's say these are sperm, and we have a normal egg that has 23, we'll do humans, it has 23 chromosomes in it. If this sperm fertilizes the egg, this sperm has 24 chromosomes in it. So we're going to have a baby that has an extra chromosome, and that's why it would be a trisomy. However, if one of these sperm fertilized the egg that had 23 chromosomes, these guys only have 22 chromosomes. So that means one of the pairs is missing. So that's where you would get a monosomy from. Okay, so moving on, it says, means there are 3 of one chromosome or 1 of one chromosome when there should be pairs of chromosomes. So kids that are born with trisomies have 47 chromosomes total. Kids that are born with monosomies have 45 chromosomes total. And we're talking about humans right now, but this can happen in any species. It's not just humans that it's taking place. So go ahead and take a look at the karyotype. This is, if you don't know this already, this is a photograph of someone's DNA. It's a real photograph where a genetic counselor has arranged the chromosomes by size. Oftentimes this is done, it's done many times. If somebody wants to go through genetic counseling because they know they have a genetic disorder that runs in their family and they want to have somebody take a look at their chromosomes. But it's also done sometimes when there has been a birth defect or a baby has miscarried. So then they take a look at the chromosomes to try to figure out what actually happened. So in this case, the baby probably miscarried. It's called trisomy 18. And it is a trisomy that causes massive birth defects. And kids who have trisomy 18 almost always pass away. And then would it have been a male or a female? This was a female baby because it has 2 X chromosomes and no Y chromosome. Okay, let's talk about some of the most famous monosomy and trisomies. So Down syndrome is one of the only survivable trisomies, and that is because the chromosome 20 number 21 is really, really small. If a baby's born with one of the larger chromosomes and they have 3 of that, that's almost always a fatal condition. It causes severe, severe birth defects. And for human beings, that is just not a survivable condition. But trisomy 21, just a tiny little chromosome, and so that ends up being survival. Down syndrome has very specific symptoms because of this extra chromosome. So it causes mental disability. It causes very distinct heart defects. It also causes very distinct facial features and body features. Things like slanting eyes, a flat head on the back, low set ears, very, very common and quite easy for even the average person to recognize those particular symptoms. Okay, so they're also called, Down syndrome is called trisomy 21. So take a look at the karyotype and make sure you can identify. And was this a boy or a girl? Was a boy. All right, the next one is called Klinefelter syndrome. Oh, I'm just covering up my fruit fly. Okay, Klinefelter syndrome. These are males that are XXY. So take a look at the karyotype and see if you can see the XXY. So these boys, nothing wrong really at all. It's an extremely survivable condition. That's because one of the X's ends up shutting down, just like it does with females. So it becomes a bar body. So it's not a problem. However, it does release for the male additional levels of estrogen. So when these boys who are in every way normal, when they hit puberty, they start to develop what are called secondary female sex characteristics. Now, that doesn't mean they become a girl at all. Doesn't mean that at all. They feel like boys. Everything about them is boy. Everything about them is boy. They don't have a uterus. They don't have ovaries. They can't have a baby. Nothing like that. However, the extra estrogen produces breast development, causes them to have less facial hair, their voices tend to not deepen, they might have less musculature than maybe their male peers do who are going through puberty. And unfortunately for these boys, I mean, there's really not much. It's a very survivable condition, but they are infertile. Typically, they're infertile, which means that they can't father children of their own. But with breast reduction surgery, they can often have that extra breast tissue removed. And then with treatments with testosterone, they can improve their testosterone levels, and often lead completely normal lives, and no one would have any idea. It is an extremely underdiagnosed genetic disorder. So they think about 1 in 500 males has Klinefelters. So that would mean at our high school, 1 to 2 boys has Klinefelters. All right, and then as you're looking at the karyotype, what else do you notice? And hopefully you notice that this person also has trisomy 21. So this is a person that has Klinefelters and Down syndrome simultaneously. All right, and then this last one that we're going to talk about, the syndrome, is called Turner's syndrome. These are females that are XO, and what the O means is empty set. So they didn't inherit that second X chromosome. It's very, very mild condition. I'm guessing very underdiagnosed also. One thing that is sort of a physical manifestation is that they have what's called a webbed neck. Kids are on, when students hear that, they're like, oh, what? It's just a wide neck. Most people would have no idea that they have a webbed neck. Anyway, there are some other impacts internally, but otherwise the girls are normal. They tend to be shorter than average, but unfortunately they are also infertile. So beyond that, most of the impacts are quite minor. Some possible heart issues, and I said that they're shorter. All right, now I want you to take a moment and study this karyotype and tell me what you think about it. Okay. It's not right? If I said a trisomy is usually a fatal condition, Down syndrome and Klinefelters being like the only ones that are survivable, this individual has trisomy on every single chromosome. So this is actually called Triploidy instead of trisomy. It is a fatal condition in humans, and it's due to one egg being fertilized simultaneously by 2 sperm. So those 2 sperm are carrying 23 chromosomes each, and then the egg has 23 chromosomes. So this individual would end up with a total of 69 chromosomes, and that's always a fatal condition in humans. What's interesting is that polyploidy is actually a way of life for some organisms. So there's a high incidence of polyploidy. Poly just means more than 2 chromosomes. There's a high incidence of polyploidy in plants, frogs, and fish, and it appears to give them some kinds of advantages. So commercially, we've been making the most of these polyploid animals because, now this is a quote that I found in my research, they have improved growth. So we would be talking about like clams and certain varieties of fish and whatever, things that are grown commercially. They have improved growth, better pathogen resistance. They tend to be infertile, which sounds bad, but that's actually good. Trixie, sorry, my doggie is barking. If she barks too much, I'll have to stop the video. All right. They are often infertile, which sounds bad, but for the commercial industry, they actually like that because it preserves their specific genetics that they're trying to go for, the genetics that they like of their particular commercial food. Okay. And then it says, what are the advantages? It appears to prevent the accumulation of recessive mutations. Listen to this because it's pretty cool. So it seems to prevent the accumulation of recessive mutations in future generations, leading to hybrid vigor, which means that these polyploid organisms are more healthy. They're doing better. And the question is, well, why is that? It's deadly in humans. It appears that these polyploid organisms have a similar benefit that females have over males by having that backup X chromosome. So if they have 4 chromosomes and one of them is mutated and not working so well, they've got a bunch of backups that will take care of them and help them to be a healthier organism. So polyploidy in humans doesn't work at all, but in some species, polyploidy is a great adaptation. All right, last section that we're going to talk about is chromosomal mutations themselves. There are four major things that can happen to the chromosomes. And just take a look at the diagram because they are super easy and super self-explanatory. Number one, part of a chromosome can get deleted. It can get broken off, damaged, whatever, but part of it is gone. It can be duplicated. So notice the BC genes get duplicated. We saw that happening in proto-oncogenes during cancer. Sometimes proto-oncogenes are accidentally duplicated, so we have several proto-oncogenes. Proto-oncogenes increase cell division, and so that leads to cancer if you have too many proto-oncogenes. So that's called a duplication. And inversion, to invert something means to flip it over. So take a look at how it was BCD in the original chromosome, and now it is DCB. So that can cause any number of effects. The genes are still there, but the order in which those genes are supposed to be read is backwards. May or may not have an impact on the organism. And then the last one is called a translocation. The translocation is where one chromosome breaks and then it actually, no joke, reattaches to a different chromosome. So notice that the AB has broken off the original chromosome and now it has attached to this other one over here. And they've translocated in that circumstance. So just last thing, some of these rearrangements are detrimental, even deadly, and sometimes they're silent, which means that the person who has these different effects has no idea they have them, and oftentimes they don't appear until they've had offspring, and the offspring inherit some of these. So take a look. There's a normal chromosome 9 and a normal chromosome 2. There's a translocation, and it can lead to a type of leukemia. That's an example. This map shows a whole bunch of different translocations and different genetic diseases that are associated with these translocations. Remember, a breaking of one chromosome and a reattaching to the other, but sometimes completely harmless. All right, everybody, that's it. Let me know if you have any questions. Have a great rest of your day.